Hello everyone and welcome to this channel. In this video we will discuss about microscope. As a science graduate or a student, you must know about microscope, its function and properties. Microscope can be used to observe small objects that are too small to be seen by the naked eye, even cells. If you are new to this channel, do subscribe this channel quickly. Click on bell icon so you may get notifications and updates. Despite some early observations of bacteria and cells, the microscope impacted other sciences like botany, zoology more than medicine. Before the invention or introduction of microscopy technique, human beings didn't know about the tiny, small, invisible creatures known as microorganisms. Do you know, in the late 16th century, several Dutch lens makers designed devices that magnified objects. But in 1609, Galileo perfected the first device known as microscope. Later in the 16th century, Anton van Leeuwenhoek began polishing and grinding lenses when he discovered that certain shaped lenses increased an image size. The glass lenses that he created could enlarge an object many times. The quality of his lenses allowed him, for the first in history, to see the many microscopic animals, bacteria, intricate detail of common objects. Leeuwenhoek is considered the founder of the study of microscopy and played a vital role in the development of cell theory. To understand the microscopy, one must know the difference between magnification and resolution. Magnification is the process of enlarging something only in appearance, not in physical size. Or we can say it is a process of zooming in and out of the picture or image. While resolution is the finest sharpness and clarity of the produced image when viewed through an optical. In image resolution describe the image level of detail. Higher resolution means more image detail. It is the ability of the instrument to produce a detailed image of a subject. There are several types of microscopes available in the market, but the most common types are mentioned here. Starting from number 1 is compound microscope, confocal microscope, stereoscope or dissecting microscope, scanning electron microscope, transmission electron microscope. We will discuss here compound microscope because this is the most common type. Compound microscopes are light illuminated. The image seen with this type of microscope is two-dimensional. This microscope is the most commonly used. You can view individual cells, even living ones. It has a high magnification, however, it has a low resolution. Let's talk about structure of microscope. You can see labeled microscope diagram, where you can see its parts very clearly. In this video, I am going to share the details of parts of a compound microscope, from which you can learn very well. Let's start from the top of microscope. On top, we have oculars. We can adjust as out or in to fit the width of your eye. The ocular have a magnification of 16x or maybe 10x. Below that, you can see the nose piece. This is the revolving nose piece. Each of the lenses you see on the revolving nose piece has a different magnification. This microscope is outfitted with 3 objectives, a 10x, a 40x and a 100x. Below the nose piece is the stage. On this stage there is a clamp for holding the slide. In the center of the stage there is a hole, and underneath this hole is the condenser. So if we look underneath the stage, you see the condenser is up here. The condenser can be controlled and moved up and down, but for most applications all the way to the top is the best position. On the condenser there is a small lever. This is the iris diaphragm lever, and it controls the amount of light that passes through the condenser. The light intensity can be controlled directly below the condenser. On the front of the microscope there is a switch which will turn on the light. Below the stage on one side there are the stage moving controls. One control moves the stage back and forth. The lower control moves the stage right and left. These dials move the stage up and down to help focus of specimen. The coarse adjustment moves the stage much smaller increments. When you finish using the microscope, make sure that you clean it. To clean it, you will use lens paper. Don't use tissue paper or cloth. Use lens paper to wipe off the oculars, and you can also wipe off the objectives. If you used oil on your microscope, you want to use the lens cleaner to clean off any oil. Just put a couple of drops on your paper and run to remove any oil. Hope you liked this short detailed video. Thanks for watching.